This video provides an overview of a set of tutorials on RNA sequencing data analysis. These tutorials were created by a team here at the Graduate School of Biomedical Sciences and Engineering at the University of Maine. RNA sequencing is a powerful tool to allow us to study genes and genomes. RNA, of course, are transcripts. When a gene is expressed, a transcript or a mature messenger RNA is expressed. And RNA sequencing allows us to identify the sequence of that entire transcript. RNA sequencing also allows us to estimate the level of expression of every gene that's expressed by a given cell or tissue. And the idea is that the number of sequencing reads generated from an RNA sequencing experiment is proportional to the level of expression of those RNA molecules. So RNA sequencing involves taking the RNA that's extracted from tissues or cells, shown on the left. That RNA is processed to remove ribosomal RNA, because most RNA in a total RNA sample are ribosomal RNAs. Those RNAs are fragmented, cDNA is synthesized, and then adapters that are specific to the sequencing platform are ligated onto the ends of those cDNA fragments, and then finally those cDNA fragments are sequenced. And for a given sample with bulk RNA sequencing, which is where you're characterizing expression in a tissue or a large number of cells, you might generate 30 million sequencing reads per sample. And on the right, this image is trying to illustrate how the individual sequencing reads, which are those short rectangles, how those align to, in this case, two different exons of a zebrafish homeobox transcription factor named HOXB1B. HOXB1B in the zebrafish just has two exons. Exon 1 is actually on the right because this strand is on the negative sense of the genome, negative strand of the DNA. Then exon 2 is on the left. And again, the level of gene expression of HOXB1B is proportional to the number of aligned RNA sequencing reads. One application of RNA sequencing is bulk RNA sequencing, where you're characterizing gene expression in a set of tissues or a large number of cells. And one application of RNA sequencing is just to characterize all the genes that are expressed in a given tissue or cell. And you can determine different isoforms that are expressed in that tissue, or just what are all of the genes that are expressed in that tissue, and that can be important. A more common application of bulk RNA sequencing is to study how gene expression changes with different treatments. And in this box, we're illustrating one of the data sets that are featured in our tutorials. A research group at MDI Biological Laboratory here in Maine had used zebrafish as a way of modeling chronic stress. And they were interested in how exposure to chronic stress during embryonic development may alter the expression of genes in adult animals. And in this case, they were using zebrafish as a model for, and so on the left, we have an illustration of our control group, where the animals were exposed to DMSO during the first 5 days of development. Then on the right, the treated group, they were treated to 1 micromolar cortisol for the first 5 days of development. Then those fish were raised under clean conditions until they were 1 year old. And at that point, blood samples were taken. And from those blood samples, RNA sequencing was performed. And in this case, they had 3 biological replicate samples. Another application of RNA sequencing is to profile microRNA expression using an approach called small RNA sequencing. And microRNAs are short non-coding RNAs, approximately 22 nucleotides in length, that negatively regulate different target genes. You can use small RNA sequencing to profile microRNA expression in different tissues or cells, and then also look at, in other experiments, how microRNAs are differentially expressed with a given treatment or set of treatments. One of the small RNA sequencing data sets we include in our tutorials is from a study of heart regeneration in zebrafish. Zebrafish have an amazing ability to regenerate their heart or cardiac tissue approximately 30 days after injury, and where you cannot tell that the heart was originally injured. So in this tutorial, again, small RNA sequencing in the zebrafish tutorial, we compare expression of microRNAs in a control group, which is at the initial point of injury of the heart, and then expression of microRNAs 3 days later, and then look at the expression of different microRNAs. With different RNA sequencing data sets, there is a basic analysis workflow that consists of doing some quality control diagnostic analyses of your reads, as shown on the left, and then mapping those reads to a reference genome or transcriptome. And as part of that process, analyzing those aligned reads and then estimating or counting how many reads map to the exons of a given gene. And so we're going from sequence reads on the left to read counts on the right. Once you have those read or gene counts as labeled in this diagram, then one can do statistical analysis of those reads and determine which genes are differentially expressed. Then one can also take those differentially expressed genes and do different types of analyses. And in this case, we have a tutorial that features how to analyze transcription factor co-expression analysis using a package called NetAct. Now that you're familiar with the general analysis workflow, we wanted to explain the different tutorials and how they map to that workflow. We have a set of tutorials that walk you through doing analyses using data generated from the mouse, as well as a set for the zebrafish and then for C. elegans. And these are 3 models that are widely used among researchers here at the Graduate School for Biomedical Sciences and Engineering at the University of Maine. The first set of tutorials covers read mapping and generating the read counts. Tutorial 2 reads in those read counts and tests for differentially expressed genes using DE-seq2. Another tutorial demonstrates how to use the NetAct package to analyze transcription factor networks using data from the mouse. Tutorial 3 is a small RNA sequencing analysis workflow that begins with reading in the small RNA sequencing data, annotating microRNAs that are expressed in the data, and then doing the differential expression analysis, again using DE-seq2. Tutorial 4 is a single-cell RNA-seq analysis workflow that uses the Seurat package. This is the list of the mouse workflows that we've put together. We have a set for bulk RNA sequencing and then small RNA sequencing to look at differentially expressed microRNAs, as well as single-cell RNA-seq. For bulk RNA sequencing, there are actually 4 different tutorials. 2 of them, tutorial 1 and 1b, deal with read pre-processing and mapping in order to get those read counts. Then tutorial 2 is analyzing those read counts with DESeq2 to find differentially expressed genes. And then tutorial 2b is also analyzing the read counts, but it is using that NetAct package to look at transcription factor networks. The difference between the first 2 tutorials, tutorial 1 and 1b, is the size of the data. So read mapping is computationally intensive, and so we wanted to have a tutorial, which is tutorial 1, that runs very quickly, where you're analyzing a very, very small subset of the data so that the tools run very quickly and you can learn how to run the tools. Tutorial 1b is the full data set, where you're downloading approximately 30 million reads per sample from the sequence read archive at the NCBI, and you're analyzing the entire data set. That takes much longer to run. For small RNA sequencing, there's just a single tutorial that covers all of the read processing as well as the differential expression analysis. And then finally, the tutorial 4 that's analyzing the single-cell RNA-seq. For zebrafish, it's similar in that we have bulk RNA sequencing workflows, small RNA-seq for microRNA expression profiling, as well as single-cell RNA-seq. And like the mouse, we have a read mapping tutorial, tutorial 1, that runs very quickly but uses a subset of the data. And then tutorial 1b that uses, again, approximately 30 million reads per sample. It's the entire data set, and it takes much longer to run. We don't have a NetAct tutorial to look at transcription factor networks in the zebrafish, but the rest are here. For C. elegans, the roundworm, we have bulk RNA sequencing as well as small RNA sequencing tutorials. And like what we did with the mouse and zebrafish, we have a tutorial that runs very quickly on a subset of the data, as well as one that works on the entire full data set. So hopefully you'll find those species-specific workflows useful. So in summary, we have these sets of tutorials that cover how to analyze RNA sequencing data that's either generated with bulk RNA sequencing, small RNA sequencing, or single-cell RNA sequencing. So we hope you find these useful, and if you have comments or suggestions, please reach out to us. I'd like to acknowledge the team here at the University of Maine who worked very hard on putting these tutorials together, as well as our colleagues at the NIGMS Sandbox who provided instrumental help along the process. And finally, our funding through the NIH NIGMS T32 grant that we have here at the University of Maine. Thank you.